Hello students, welcome to SK Biology. Today's video is third part in which we are discussing discovery of genetic material. In the quest of genetic material, in last video we learned transformation experiment of Avery, MacLeod and McCarthy, in which they have proved that genetic material is DNA. After this direct evidence, there are still some scientists who believed that protein is more complex in nature than that of DNA. So, heredity is carried by protein. This confusion of scientists raised question that genetic material is DNA or protein. This mystery was solved by scientists Hershey and Chase in 1952 with simple but powerful experiment. Let us understand their experiment. For experiment, Hershey and Chase selected a bacteriophage, virus that infects bacteria. The important feature of this bacteriophage is it contains only two biomolecules, DNA and protein. DNA present inside of bacteriophage and protein present outside in the coat. In DNA, phosphorus is present and in protein, sulfur is present due to sulfur containing amino acid. DNA does not contain any sulfur atom and protein does not contain any phosphorus atom in it. This feature made bacteriophage a perfect choice for experiment. After selection of bacteriophage, Hershey and Chase labeled these bacteriophages with radioactive isotope P32 and S35 in two separate reactions. In one reaction set, they have used medium containing P32 isotope and grown bacteriophages into this medium. And in second reaction set, they have used medium containing S35 labeled isotope and grown bacteriophages into this medium. As a result, all new bacteriophages found in P32 medium will have P32 labeled DNA, that is radioactive DNA. And all new bacteriophages in S35 medium will have S35 labeled protein coat, that is radio labeled protein. In the next step of experiment, Hershey and Chase infected E. coli bacterium with these two radioactive viruses in separate reaction. After infection, to check radioactivity present in new bacteriophages, they have agitated it in a blender and centrifuged. The result they got is, in case of DNA labeled reaction, new bacteriophages are radioactive and radioactivity is present in inside cell. In case of protein labeled reaction, new bacteriophages does not have any radioactivity. Radioactivity was found in medium. This result proves that only DNA enters bacterial cell and directs the formation of new viruses, not the protein. Hence, Hershey and Chase experiment confirmed the result of Avery, MacLeod and McCarthy that genetic material is DNA.